Um, my name's Howard Rundle. I'm a professor at University of Ottawa and the executive vice president of the Society for the Study of Evolution. It's my pleasure to moderate this session. We've got 4 speakers lined up, although unclear one. We're still waiting on one to show up. We'll stick to the schedule either way. And with that, I'd like to turn things over to our first speaker, Jordan Gossett. Take it away.
Hi, yeah, my name's Jordan Gossett. I'm a PhD candidate at the University of Hawaii at Manoa, and today I'm going to be talking with you all about rapid diversification and cave-adapted plant hoppers across Hawaii's volcanic landscape, which is some work I've been doing as a part of my dissertation. So just to get started, Fulgoromorpha are more commonly known as the plant hoppers, is the model system that I study. They're a diverse group of sap-feeding insects that can serve as a model system for studying rapid diversification and speciation for several reasons. Within this group, the family Suxiidae represents a disproportionately high share of plant hopper diversity, largely due to elevated rates of endemism. Many succeeded lineages have independently colonized novel environments, including repeated transitions into subterranean cave systems, and this provides us with natural replicates for examining how ecological opportunity may drive diversification. Additionally, plant hoppers maintain obligate endosymbiotic relationships with bacteria that help supplement essential nutrients that are missing from their plant sap diets, and really strong coevolution between plant hopper hosts and these endosymbionts can induce rapid ecological and genetic changes in plant hopper hosts. Altogether, these traits make plant hoppers an ideal system for investigating the mechanisms that promote diversification across multiple scales, including ecological, geographic, and temporal. The Hawaiian archipelago is a known biodiversity hotspot, and within the archipelago, I'm working on Hawaii Island, which is the youngest island. It's estimated to be only around 500,000 years old, and its volcanoes are still actively shaping the landscape. Following each volcanic eruption, lava tube caves are formed and make up an island-wide subterranean network of cave habitat. And so you can see on this image, all of the different striations and colors represent different lava flows that are kind of layered one on top of the other in different volcanoes. Within the succeeded plant hoppers, a subset in the genus Olearis have migrated underground and become fully cave adapted. The cave adapted variant in Olearis that's most common is known as Olearis polyphemus. And as you can see here, these cave adapted plant hoppers show classic troglomorphic traits such as reduced eyes, reduced wings, lack of pigment. And this was once thought to be a single widespread species of Olearis polyphemus. However, within the past 20 years or so, some research has come out observing distinct mating signals between plant hoppers that are located in different caves that are within very close proximity to one another. And on top of this, there's also been observations of really strict assortative mating based on these vibrational signals, which suggests that Olearis polyphemus might actually be a cryptic species complex. And so my dissertation work focuses on how this phenotypic and behavioral divergence may correspond to genetic differentiation across the island's subterranean habitat to help resolve this question. So frequent volcanic eruptions create these lava tube caves, and we get this sort of ecological succession where new habitat is layered on top of old habitat that eventually degrades. And so we not only get this fragmentation where habitats kind of stacked on top of one another, but we also get fragmentation within lava flows and within caves, as you can see with this cross section of a lava tube cave here. And this fragmentation likely limits gene flow and has potentially promoted rapid and repeated divergence in Olearis polyphemus. In terms of looking at divergence on a temporal scale, we're really lucky in this system. It's a young system, so we're able to easily date geologic events. So we know in terms of age of the island, we know that Kohala is the oldest volcano, Mauna Kea is next, Hualalai after that, Mauna Loa, and then finally Kilauea is the youngest volcano on this island. And Because each volcano and thus each lava flow can be geologically dated, we know the timing of cave formation events for these habitats, which helps us calibrate the chronology of historical processes, such as colonization and divergence in Polyphemus. So in terms of looking as the preliminary view at genetic or genomic variation in Olearis polyphemus, I did a little bit of work earlier on in my PhD looking at how host-symbiote coevolution may impact divergence in Olearis polyphemus. And at a broad scale, I looked at comparisons of endosymbiont genomes between surface hosts and cave hosts, and we did see differences in endosymbiont genomes at that level of comparison. But what's more is that when we looked between two Olearis polyphemus hosts, we saw similar levels of genomic variation in their endosymbionts, indicating that at a really shallow timescale, there's a lot more host divergence in terms of coevolution than we would expect to see at this level. And so that gets at this larger question of, are there genetically distinct lineages of Olearis polyphemus across Hawaii Island? And so in order to tackle this question, I have collected 110 Olearis plant hoppers for DD Rad Seek. About five or six of those are surface plant hoppers, while the remainder are Olearis polyphemus cave-adapted plant hoppers. I have a library of 1,300 SNPs that have been assembled for downstream analysis. In terms of where my samples were collected from, I'll just orient you a little bit to the time scale we're working at. And I'll map on this image here that shows our sampling sites. And all of the highlighted flows are where we have samples from. And I highlight this onto this previous image to show you that while I'm highlighting certain sampling sites, they're still interconnected at their core. And so we have samples from 3 volcanoes, Hualalai, Mauna Loa, and Kilauea. Within Hualalai, that is our oldest volcano that we've sampled. We have samples from a flow that's around 3,000 years old, this QH2 in purple. From Mauna Loa, we have samples from 3 flows, QK5 up top being the youngest, QK3 and QK2 being anywhere from 750 to 3,000 years old. And then from the Kilauea volcano, we have samples from the QP4 flow, which is another really young flow. And so what you can see just based on these ages is that we're dealing with extremely young habitat in terms of sampling here. And so on to my first result. So in order to understand evolutionary relationships of Olearis polyphemus kind of as a broad view across the island, I constructed a maximum likelihood tree using my SNP assembly. Up top here, we have our surface-dwelling plant hoppers that are used as an outgroup, and everything below that is Olearis polyphemus. All of these lot numbers represent an individual. And I think sorry, let me go through the flows really quick. So up top, we have a clade from QK5 in northern Mauna Loa. We have a clade from QP4 in Kilauea. We have a QK3 clade, a QH2 clade in Hualalai, and another QK3 clade. And so I think the biggest pattern to immediately recognize here is that most lava flows form these clear monophyletic clades, with the exception being QK3, which forms 2 polyphyletic clades, suggesting maybe some intraflow divergence. And those are both located, all of the samples from these clades are located within really close proximity within this lower QK3 region. So to investigate this further, I ran the SNAP program in BEAST to create a consensus species tree. And while the overall pattern is slightly different, the main finding is that there is still strong support for a split between those 2 QK3 clades. And I'll now refer to those as QK3A and QK3B. And just thinking a little bit about the geology of this area, I've layered on the map of the rift zone that kind of transverses the entire island. And so QK3 does lie within a rift zone, which is an area where there's increased volcanic activity, which could potentially increase vicariance events and lead to rapid divergence, even within flows in this really dynamic area of the island. And to look at this a little further on a more fine scale in terms of population genetic level patterns, I ran a PCA plot and an admixture grouped by flow, individuals from different flows. And so as you can see in the PCA plot down at the bottom, each cluster represents a different flow across the island. And the main pattern together here is that there's pretty distinct clusters with minimal overlap between them, with the exception of a few individuals. Whereas when we look at admixture, this indicates we've got these kind of distinct clusters that we're seeing shared ancestry across different flows across the island. And there's low admixture, but not complete isolation. And so these patterns may be reflective of incomplete lineage sorting or recent introgression, which, when we think about it, is pretty plausible given what we're dealing with in this system. It's a really young system. It's under 500,000 years old. There has been repeated bottlenecks of plant hoppers during those underground transitions, as well as frequent cave colonizations and frequent habitat turnover due to the volcanic activity. And so these patterns kind of reflect the complexity of the system, I think. And so to look at this idea of, okay, is this introgression? Is it incomplete lineage sorting? I ran a few analyses to try to get at that. The first was a tree mix analysis, which basically just infers historical admixture events. And the results show a few things. One, we seem to have allele sharing from surface into this QH2 flow, which is our oldest flow that we have sampled, our oldest volcano that we have sampled. And we also see some interesting, with the orange arrow and the blue arrow, it looks like some gene flow in and out of and between QK3. So maybe some hybridization in this zone. To look at this further, I ran D-Suite, which helps tease apart introgression versus incomplete lineage sorting. And on the right-hand side, you can see kind of like the heat map key. That's the FB estimate, which indicates the proportion of the genome that is being shared due to introgression between a particular population on the top and a branch of the tree on the left. So if this were incomplete lineage sorting, we would expect kind of an even gradient across the grid here, but instead we see these localized hotspots of elevated gene flow kind of within and between QK3. These findings suggest that populations in this region are in the process maybe of divergence or potentially experiencing secondary contact, with localized introgression maybe pointing to incomplete barriers to gene flow on a really fine scale within one single lava flow on the island. But there's still a little bit of work to do to kind of tease this all apart. So just in conclusion, what do we know at this point in terms of, is this divergence, is in secondary contact, what's going on? Within the QK3 flow at the tip of the island there, we do know that there is variation in host symbiont coevolution. So going back to one of my first intro slides, when I talked about my previous work in my dissertation, we did see within very close proximity two hosts in this QK3 flow show divergence in their endosymbiotic genomes. We also see some potential morphological variation. Miri Steck at UH Manoa in my lab is working on this. We don't know a lot about it now, but it is something we want to look into further. And also within this flow, we do see distinct mating calls and assortative mating. And so these processes are commonly seen in the first stages of divergence or reinforcement and support isolation within QK3 and point to potential reproductive barriers, which is really cool because in this young system, we're essentially testing and looking for these evolutionary processes as they're actually occurring in real time. And so to be able to understand patterns and directions of colonization between lava flows and to explicitly test for divergence out of QK3 or secondary contact within the zone, my next step is to apply approximate Bayesian computation models to formally test these ideas. And thank you. That is all I have.
Thank you. If you could stop sharing your slides, and Suzie, you can go ahead and share yours, but we have time for a quick question if there is one from the audience while we prepare for the next one. Feel free to use the Q&A function of the chat or raise your hand and I can unmute you. If not, I'm curious. You mentioned that this coevolution with endosymbionts could be a mechanism to drive speciation in this group. Is there actually evidence that reproductive isolation has resulted from this coevolutionary process unfolding in different ways and different caves? Is that a hypothesis at this point, or is there evidence suggesting?
Yeah, so you actually hit on what I'm doing, what I'm going to do for the third chapter of my dissertation is actually test that explicitly. So for the work I talked about, we only have two cave hosts, and so it's not enough to definitively say. But the fact that between just two random hosts that we collected, that we did see that divergence in their endosymbionts suggests that maybe on a larger scale it does play a more significant
Cool.
role. So
I
yeah.
guess we should, sorry, I took long asking my own question. We should keep moving. Suzie, are you able to share your slides? I saw you in the, yep.
Yes, hello. I'm having trouble sharing my slides. So if
Uh-oh.
sorry, just one. um okay, I think I can do it this way. Okay, sorry, I don't think
If you're unable to get it right now, then you can reach out to the organizers. I know they emailed you, and there may be the
yes,
possibility of
okay.
rescheduling you in an empty slot in a later session.
Okay, yes, I'm so sorry. Yeah.
So, yeah,
We could also try if your talk is fast, we could also try emailing me your slides right now and I can call them up. But otherwise we will try and reschedule for tomorrow.
Okay, yes.
we're almost 2 minutes
I'll
in already
email
now.
you.
Yeah.
Yes, I'll email you right now and then I'll
Okay, we'll reschedule. Sounds good.
Well, in that case, I'll just remind the audience that we're going to keep the schedule because sometimes people want to move between sessions. And so we'll continue with the third talk at starting at 2 o'clock. Exactly. And if there's any other discussion on Jordan's talk, we've got a little bit of time to do that. Jordan, I was surprised if you're still there.
Yep.
Well, maybe not surprised, but I don't know anything about the system. It sounds like a really neat system, but from your phylogeny, it indicates there's a single evolutionary origin of these cave species, and then they've spread to all the different caves you sampled, as opposed to being multiple origins from the above ground species. That seems a bit surprising ecologically that if you're adapted to caves, there's a lot of intervening inhospitable habitat from colonizing one cave to another. Do you have any idea how that's occurring? How are they moving?
Yeah. It's a really dynamic system in that way. We do know that plant hoppers are pretty good at dispersing once they're underground. And the thing about the system is the lava rock, it's super porous. And so, a plant hopper, the ones I study are like you could fit three of them on your pinky fingernail in some cases. And so we know that they're good at dispersing maybe through cracks and things that as humans we're not seeing as maybe passages to dispersal.
So you think they're dispersing underground among caves? Because you've got volcanoes on different sides of the island, which are
Yeah,
pretty distant.
they're pretty distant. Yeah, and it's hard to tell on some of the maps, but everything is very connected. Even flows that have come from different volcanoes can be connected to one another. So, yeah, we definitely think they're dispersing underground.
I was wondering if I could ask a question.
Yeah.
Yes, hi Jordan. I'm afraid I missed part of your talk, and I was just curious about your PCA slide, your figure. Do you know what PCO and PC1 kind of represent?
Yes, that's actually something we've been trying to conceptualize. We think it might be kind of like length and width of the island, spreading across different flows. But I'm not entirely I haven't really nailed that down just yet, but we think it's probably like dispersal from the older side of the island to the younger side of the island.
Okay, because because just length and width would not necessarily mean anything, right? So there is some other variable there, which is the age of the island. Okay, that
Yeah, yeah,
that
we
I,
think
I
it's from the one side to the other potentially.
ah, all right. I was just curious because you, there used to be a time when people would, when they show the PCA, figure, they would actually write what PC1 and PC2 probably are, but I've almost never seen that lately. It's always PC1 and PC2, and I'm always curious. The whole point of that is to try and understand how these things vary across two variables and two sort of composite variables, and so that's why I was unmeasured variables. So that's why I was curious about that. No, I'm glad you're thinking about that and speculating on that. No, thank you. Appreciate it.
Yeah, for sure. That's a really good point.
So they're using vibrational mating calls. Is that correct?
Yeah, that's
But
correct.
are they on plants in these cave systems? Like maybe I'm envisioning the wrong thing. Maybe it's the openings that, like I would think vibration wouldn't transmit on lava rock.
Yeah, you're right. So there's plants that kind of settle on the surface of lava tube caves, and they project their roots down through the cracks into the caves to get moisture and nutrients. And so you kind of have all these root curtains that are hanging.
Okay, and they're
And
on the
so
roots.
they hang out on the roots, and then
Oh,
they
cool.
project, through the roots.
It'd be nice to see it. A
Yeah.
photo of a cave system actually in your talk. That's neat.
I'm just gonna break in quickly here. Suzie, if you're still here, we can actually, thinking about it, we can actually fit you in after, there's only 4 talks in this
Oh yeah,
session.
that's right. Yeah.
So we
Yes,
If
can
you
fit
can
you
sort
in
out the
after
sharing
fourth
now, yeah.
speaker.
yes, I
So
just yeah, sorry.
perfect. You just sent me your slides. Okay, I'll figure it out in the meantime then.
Yeah, so we'll just
Thank you.
update to the schedule. We'll the last 2 slots were empty, so we'll shift Suzie to the right after what was scheduled as the final speaker, Martina. So at 2:30 we'll
Yeah,
go with
if
Suzie.
Karen, if you want me to share the screen, I'm able to do that. So maybe I can share Suzie's presentation as well.
I think whatever works.
I'm just
So
going to
we'll
disappear for
we'll
30
figure it
seconds
out
here.
then.
I'll be right
Yeah.
back.
And I'm gonna pause the recording for a moment.
Jesús Ernesto will be speaking about asymmetric reinforcement in damselflies. I'll turn it over to you.
Okay. Good morning. Good afternoon, everyone. My name is Jesus. I am a PhD student in Ecological, Mexico. And this is part of my PhD dissertation. And well, let's start by defining reinforcements. Reinforcement is the enhancement of pre-zygotic isolation in sympatry by means of natural selection. Reinforcements have some clear consequences. First, in terms of reproductive isolation, it increased the reproductive isolation in sympatry, but it can also generate reproductive character displacements in the traits involved, related with the barrier that is enhanced. Reinforcement have some clear predictions nowadays. First, it predicts that the increase in sympatry of the isolation. It also predicts a higher and quicker isolation in the rarer female when these species are with different proportions in the population. And it also predicts that we have not only find higher prezygotic isolation, but also lower prezygotic isolation in sympatry after reinforcement. In polymorphic species, we can expect more pronounced partners of asymmetries related with alternative reproductive strategies, for example, related with associative mating and mate recognitions. Let's take this example. Let's suppose we have two species with two polymorphic species when one of the morph is similar within the species and have some level of assortative mating within morphs. In allopatry, we can have this clear pattern of assortative mating, but in sympatry, the similar morph can get involved in heterospecific matings. This can lead to the similar morph to have lower fitness in sympatry, and this means a higher of reinforcements. So in this work, we try to test these differences within morphs using two damselfly species from Europe, from Ischnura elegans and Ischnura gaelsis. These damselflies have three female morphs, one male morph, one male, one female-like morph, and two conspicuous morphs that is called genochromes. These species are so similar that the only way to differences them is between the secondary reproductive organs, the cerci and the pronotum of the females. And this is important because during the copulation, the matings, this is the first point of contact that allows the female to recognize the male during the tandem attempt. In the last century, Ischnura elegans entered in secondary contact, expanding its range from Europe to the peninsula and formed two independent hybrid zones in Spain, one in the northwest and one in the central and Mediterranean area. During this expansion, hybridization occurs, and we have already found some consequences. First, in the northwest area, we have bidirectional integration, where we can find hybrids from different generations. Meanwhile, in the north central, we have unidirectional introgression to Ischnura graelsi, who is the endemic species from the peninsula, and most of the hybrids from the F1 generation. Also, we can find that there is a change in the morph frequency in the populations. The androchrome female, that is the one that is similar to the male, is usually the rarest in the southern area of the distribution. But in sympatry, most of the population have high frequencies of this female morph for both species. Recently, Valdez found reinforcements of the mechanical barrier. These barriers involves the ability of the male and female to involve in tandems. So he found asymmetric reinforcement in the cross involving elegant females. And these reinforcements have some consequences, have reproductive character displacements in the elegant females and graelsi males, but also in elegance males, and it was attributed to lock and key coevolution between males and females of Ischnura elegans. Why these species are so good to test for asymmetric reinforcements? First, there is some discrepancy in allopatry between the matings, the frequency in matings and the frequency of the females. So there is a tendency for males to prefer the genochrome females in the populations, but this change in sympatry where the partners breakdowns and androchrome females seems to be preferred by Ischnura elegans males. There is also contrasting alternative reproductive strategies between morph and between species. In Ischnura elegans, the genochrome females have higher fecundity. Meanwhile, in Ischnura graelsi, genochrome females have lower fecundity, but is more related with high male density in the populations. So to test this, we sampled 29 populations along the hybrid zones and allopatry for both species. And we collected 896 crosses within the last 24 years. With these crosses includes crosses in laboratory conditions and natural conditions. So in this process, we measure reproductive isolation by means of 5 barriers: the tandem barriers, but that's the ability of male and females to involve in tandems, the matings, the ovipositions, the fecundity, and the fertility. With these barriers, we estimate the absolute and the cumulative isolation by each barrier. And finally, we construct generalizing linear models and test by contrast test between allopatry and distinct hybrid zones. So what did we find? In the process involving Ischnura graelsi, we found reduced cumulative isolation in sympatry in both hybrid zones. And for both morph and in the crosses involving Ischnura elegans, we find increased cumulative isolation in sympatry for both morph. Interestingly, the asymmetric, the chain is asymmetric between the morphs. When the reduction is in Ischnura graelsi, the change is most pronounced in the androchrome females. Meanwhile, in Ischnura elegans, the increase in the insolation is most pronounced in the genochrome female. When we check by each barrier, we find that in the cases where the reduction occurs, the barrier that occasioned this reduction was the oviposition barrier. So in sympatry, more females are able to produce, more genochrome females are able to oviposite. We also find that both females have more fertility in sympatry than in allopatry. And we attribute this to changes in the hybridization, in the direction of the hybridization, and it's caused by the purge of the Peterson of Sankey-Muller incompatibilities. In the case of Ischnura elegans females, we found that the reinforcements is occasioned in the genochrome female in the two hybrid zones. And we think that is happening just in the genochrome female because the androchrome females are already isolated by means of behavior, morphology, and male preference. We also found that there is some sort of reinforcement of the fertility barrier for both morphs. And this can be, the reinforcement of these barriers can be due to low chance of evolutions of previous barriers. In the case of androchrome females, could be because there are no choices by the males. And in case of the genochrome females, maybe they are already isolated by the reinforcement of the mechanical barriers. Another explanation can be coupling of the barriers, the mechanical and the fertility barrier by means of linkage disequilibrium. So our main conclusions is that reinforcements of mechanical barriers is exclusive of the genochrome females in Ischnura elegans. This is leading to both females to be equal in the mechanical insulation in sympatry. And we hypothesize that as a cascade effect, mechanical isolation of the genochrome females can isolate this morph from males of allopatry and from androchrome females in sympatry, leading to some sort of morph speciation by cascade of reinforcements. And well, thanks to all the support I have from the institutions, the technical support, and the societies to allow me to be in this conference. Thank you so much.
Thank you. We've got a few minutes for questions if there are any from the audience. You can raise your hand or use the chat or just unmute yourself.
I guess I had one curiosity question. So how does I guess I was just curious about why females in one species and males in another species seem to have that kind of a divergent effect.
One female seems to I didn't listen the last part of the question, sorry.
Well, you showed a graph where you said that can you hear me?
Yes, please.
Okay. So let me also I didn't realize that I was also not showing myself, so maybe I should show myself. So you said that, or you showed a graph where you showed that females had a divergence in one species, whereas males seem to show the divergence in another species. And I was just wondering if you would elaborate on that. Maybe if you bring up the slide, if you could, perhaps.
Okay, let me share again. Do you have the number of the slide, by the way?
Gosh, I wish I had noted it down. It was after the, it was, I think, before this.
Oh, okay,
You showed 4 graphs where you showed line graphs. Yeah, there were 4, a panel of 4. No, line graphs.
Four.
There were a panel of 4 graphs, and the co-species were in columns.
Okay, species.
No,
we may
run out of time.
We may have to leave this for
That's
further
fine.
discussion in the chat.
Right. I'm just curious anyway. So
if you could actually stop sharing screens, or yeah, thank you. So our next speaker can go ahead and get their shared slides set up. Yep.
Okay,
Right,
I think I'm sharing.
Yep. So yeah, you are.
Okay.
So
perfect.
Welcome to our third speaker, who will be talking about ecological context and important to introgression in Drosophila hybridization events. Martina.
Okay, thank you very much. Thank you to the organizers in general for this opportunity. And I would like to thank to Universidad de Buenos Aires, where I am doing my PhD. So I'm going to talk about the ecological context that drives introgression's outcomes in experimental Drosophila hybridization. So understanding the radiation events that happened in a group could be a little bit difficult sometimes when we don't have enough information about the ecological context in the past, or when we don't have information about the genetic outcomes that have been present during their history. But in the case of Drosophila, we do have a lot of previous studies where we have information about their genetics, their physiology, their morphology. And in the particular case of the Pusati cluster inside the Drosophila species, we also have information about their ecology, as we know that they use cactus, and in particular their necrotic tissues, to breathe and to get food. So it is studied that their speciation events inside the cluster are associated with the host that they use for that. In particular, in here we are seeing their distribution, the distribution of the different species inside this cluster, which are well defined. As I was saying, these necrosis of the cacti are really useful for these insects to get shelter, in particular when we are in arid zones where the only place where they can get the food is sometimes this microhabitat. And in particular, there are different types of cacti, but we're interested in comparing what happens when these insects use or prickly pears or columnar cactus, where the columnar cactus in general are more chemically complex and more difficult to use for these insects, where the prickly pears are more kind in that sense. Of course, the use of these different hosts could be affecting and affects some traits of the fitness and life history of the flies. An advantage of this cluster is also that that there are species in a different stage of divergence. So even if they don't share a natural habitat, we can make crossings in the lab to see the outcomes and analyze what changes there. In particular, we are going to use these two sister species, Drosophila antonieta and Drosophila kepfer, and we want to characterize the hybrids and the back crosses of these two species, and we are going to analyze if the rearing cactus where each of those lines affect their phenotypic characteristics, trying to understand also if that is related with genetic background. Where did we get these flies? Well, the two species come from different areas in Argentina. One of them, Kepfer, comes from the west, which is an arid zone. On the other hand, Antonieta comes from the east, from more humid areas. And we took both of them to make the crossings back in the lab. And we also took a species of native cactus of each of the areas, prickly pears and columnar cactus native to each of them. Back in the lab then, the crossings were made. We had both males and females from each of these parental species, and we made the reciprocal F1 hybrids using females from one species and the males from the other ones. And then we passed to the back crosses, where we took females from one of the hybrids and the males from one of the parental lines. And we made that in, again, both ways. So like this, we obtained 6 lineages where we have the parental lines, the F1 hybrids, and the back crosses. Taking these 6 lineages, we can make kind of a genetic gradient where we can say, well, one of the parental lines is 100% and the other one is 0 because it doesn't have a genetic component of that other species, and the hybrids and back crosses go in the middle like this. Each of those lineages were reared in the 4 different cacti, 2 that were prickly pears and 2 that were columnar cactus, and from the different areas. Then for each of the individuals that were obtained in each of the lineages, we studied the wings. Why the wings? Well, this trait is related both with survival and reproduction. They are involved in the dispersal of the flies to get to, for example, these different hosts that they can use, but they can also be related with courtship, as they do a courtship song to attract their possible partners. And it is known from previous studies that they are affected by the rearing cactus. To do this, we studied in particular from the wings, their shape, their size, and the fluctuating asymmetry, which is like the difference between the right and the left side of the individuals. And we did that using landmarks and the procrustes method, in which we can simplify the image of the wing into these easier images that can be overlapped to see which changes are present between one sample and the other. So what did we obtain from here? Well, in here we see at the bottom this gradient of the genotype, and the different colors of the dots represent the size of the wings reared in those different medium. What we can see with the green area is what will be the additive effect of the crossings between the two parental lines. But we see both in the back crosses and the hybrids that in some cases those sizes are out of the additive zone. So we can see some transgressive inheritance in here. This was the size, but when we want to analyze the shape, we use principal components analysis, and with the little arrows you can see what is changing in each of those components. So if we put here first the parental lines, we can see that there is some phenotypic plasticity depending on the host in which they were reared. But when we add the hybrids and the back crosses, we see again that there is some transgressive inheritance over the first principal component mostly, as there is more plasticity, but they are even going further away from the shapes that were observed in the parental lineages. In what part of the wings was this change observed in particular? Well, we analyzed and tried to compare if the changes were inside in the internal details, as that will change the way in which the flies move their wings, or if it was in the general aspect. Observing the parental phenotypes again, we see that there is plasticity between the different cacti where they are reared, and again we can see that there is transgressive inheritance when we see the hybrids, and again with the back crosses. We have all of them there, and what we can see is that the greater difference is in the internal venation of those wings. When we analyze the fluctuating asymmetry, which can be like a proxy to analyze developmental instability, and because of that, this difference between the two sides of the flies, we see that in this case, this is for one of the parental lines. We see that both for the asymmetry in size and shape, there is an effect, a difference in one of the columnar cacti, and it is the one that is not native for this species. And in the case of the other parental species, Drosophila antonieta, we see that this is observed just in size, but not in shape. And again, it is in one of the exotic cactus for this species. Well, we have here the two parental lines and the overlapping images of the previous slides. And again, well, we can see that they have like opposing graphics. And when we were analyzing the hybrids and the back crosses, one of the back crosses, the one to Drosophila antonieta, many the ones spread in one of the cacti didn't even survive. They didn't get the adult phase, so we didn't include them in this analysis. And what we can see here is in size. Again, we can see there is an effect in one of the columnar cacti, but in the shape we didn't observe something that is relevant, like to difference between them. So with all that, what information are we obtaining here? In the first place, that we have a transgressive behavior, both in the shape and the size of the wings. And this is talking about genetic architecture that is not just additive. There are other effects that, of course, are affecting this phenotype. Then that there is a higher variation in the internal part of the wings, in what we call the venation pattern. And that this phenotypic variation is also associated with the varying cacti. About the cacti, those that are more chemically challenging generate more instability. It could be both because of their chemical characteristics in general, or because the species were adapted to them or not. And well, that the fluctuating asymmetry is showing more evidence of instability in size than in shape, where in shape is not always present. With this, we can observe that this non-additive inheritance is talking about how the architecture genetic patterns are disrupted when making these crossings. And there are also interactions between the genotype and the host where these individuals are reared. And with this, we can talk in the future, or we can continue our studies trying to understand how hybridizations and introgressions were involved in the divergence history of this group, which is something that we can continue doing with a little bit more information. For this, I thank you all for listening, and I'm open to any questions.
Thank you. We might have time for a very quick question if someone has one. Maybe you could, in the meantime, stop sharing your slides so the next speaker can, which will be Suzie, can start. Is there a very quick one from the audience, or?
Let's see, maybe I can try. Did you compute the heritability at all?
Sorry, can you repeat it?
Heritability, did you compute any values of heritability?
In any character in particular, we did have some more information about this, but I'm not sure what are you referring.
Okay, no, that's okay. Thanks.
Okay, you can ask me in the questions too.
Okay, thank you. And one added talk for those who might have joined late at the end here. Suzie, whose talk wasn't quite set up for the originally scheduled time. So finally, we have Suzie Choi talking about taxonomic criteria in the Impatiens genus. Turn it over to you.
Yes, thank you. So I will be talking about my research project, which is to determine the optimal taxonomic criteria for retention of species stability in the Impatiens genus. So I will go over the background, what synonyms mean, what the criteria for species designations are, my data collection analysis, and my conclusion. So first of all, our labs is studying about trait evolution, and that just means how trait changes in a single species over time, and how that's impossible because it takes millions of years. And so instead we want to use a phylogenetic tree and show how and study how the traits evolve across related species. So a good model to study how traits evolve is the plant family Balsaminaceae. And Balsaminaceae has two genera, the Impatiens and the Hydrocera genus. And the Impatiens genus, shown in the left here, has more than 1,000 species. You can see that they have tremendous diversity in their flower shape and their colors, also in their geographical distribution, habitat. But in Hydrocera, there's only one species, and it appears to be evolutionary and ecologically static, shown on the right here. And so I also compiled a table of the differences shown here. We see that, again, in patients, we have the flowers are zygomorphic. They have a bilateral symmetry, and their fruit, when they're mature, we see that this is their fruit, kind of like a pod. When they mature, these pods burst open to release their seeds, and their habitat is very diverse. And again, they have over 1,000 species. But with Hydrocera, their flowers, they have multiple lines of symmetry, which means that they're actinomorphic flowers, and that they're indehiscent fruits, which means that they're berry-like and that they don't burst open when they mature to shed seeds. They instead just fall into the ground. And their habitat, they're very static. They can only be found in semi-aquatic lowlands in Southeast Asia and in South India. So my research project is to look at, going into my project, we see that the impatient species is very prolific, and we see that there are new species being published every year. And even in this year, we found that there have been 3 new species up till today. And last year, there were 17 new species found. And in 2023, there were 11 new species discovered. And in 2022, 19 new species being published every year. So we see that this happening every year. But with many of them being described every year, we want to ask the question, were they mistakenly described as new species? And why is that many impatiens are being later determined as synonyms? And a synonyms just means that a species that is considered the same as a previously discovered species. So it's not something new. So again, I want to show this graph showing the number of species discovered in a given year in orange, and the blue shows the number of synonyms shown in that year. But I want to clarify that the blue number of synonyms do not mean that this many synonyms happened from that many species discovered in that same year. It just means that the synonyms were to be determined in that year. It doesn't mean that from the number of synonyms discovered in that specific year, that many synonyms were also determined. So my research, I want to ask the question, why are impatiens prone to synonymization? Why are there so many impatient synonyms? So first, when a new plant species is being discovered, we want to collect them from the field, shown here. So this is Helianthus annuus, or the common sunflower. We see them, they look like this out in the field. And once they're collected, you want to compare them with the literature and the herbarium specimens shown here. And this is also same for impatiens. But impatiens are notoriously difficult to identify, because here we see that in the wild, they look like this. We see their flowers, we see their stems and leaves very clearly. And this is for the Impatiens compensis. But when you compare them to the herbarium specimen, we see that you don't really see the flowers here. And it's very difficult to say that this is what we saw out in the field. So therefore, it's easy to mistake these as different species. And when you're comparing these freshly collected flowers to the herbarium specimen, what you're comparing can be classified into 4 different types, and that's what's called the criteria. So the first one, you want to compare the gross morphology, and then the second is micromorphology using microscopes. And the third criteria is paleontology, or the study of pollen. And then the fourth is phylogenetic inference. And here I have a paper showing all four criteria used when discovering this new Impatiens species, Impatiens dominigensis. So the first criteria, looking at gross morphology, is looking at the body, the structure of the plant, such as the leaves and the bottom, or the flower arrangements, the petals, the fruit. And this is used first in identification, especially when you're out in the field. The second is micromorphology, specifically for the seeds. So you're using a scanning electron microscope to look at these smaller traits, and they're specific for seed characteristics shown here. You see their shapes, their sizes. The bottom row is in a higher power, so you can actually see the seed characteristics and the presence of the granules and their texture. And then the third criteria is micromorphology, specifically of pollen, also known as paleontology. And here you're looking at pollen specifically. So the pollen shape, the size, the texture. And here I've shown pollen attached to the back of a bumblebee to show how small they are in comparison to the seeds. And then lastly, I have molecular phylogenetic inference as the last criterion, which involves reconstructing a phylogenetic tree. And these are from neutral DNA sequences. And a phylogenetic tree infers evolutionary relationships to see how closely related or how further related species are. And out of the four criteria, this is relatively the most objective because they're using neutral DNA sequences. So in my study, the objectives are to determine if there is a relationship between the criteria used for new species designation and later the stability of species status for Impatiens. So stability just means that if it retained as a species, it was stable. And if it was determined to be a synonym of another species, that means it wasn't stable. So if there was a relationship with the criteria used and the species stabilities, which one of the four criteria were the most important for maintaining the stability of a species? So I hypothesize first that there is a relationship between the criteria used for species discovery and the stability of the Impatiens species. And secondly, I hypothesize that when combining phylogenetic inference with other criteria, it will reduce the likelihood of a species later being determined as a synonym. So to test the relationship, my dependent variable is categorical, whether it was retained as a species, so stable, or if it was later determined as a synonym, not stable. And my independent variable is also categorical, is also binary. I'm not measuring anything. It's whether if the species used the criteria or did not use the criteria. So since my variables are categorical, I will be using the chi-square test of independence to test if there is a relationship between those two variables. So I've collected, our lab tech has collected the list of species and the criteria used, and I've just finished this list of species and which criteria they have used. So the ones just represents that, yes, this criteria morphology was used, and 0 means that it wasn't used. And for the synonym, 1 means that it was later determined as a synonym, and 0 means that it was later retained as a species. But since there's these overlaps, they can't be used. So we see an overlap here, here, and here, so that we need, so this we can't use this test. There needs to be independence. So the definition for the criteria was revised. So was only morphology used, or was only a combination of 2 used, or was only a combination of 3 used, or were all 4 of them used? So we see that again, when you revise the criteria, there is no, there's independence, there's no overlap between these. But we see that some of them don't use the criteria. These zeros, some impatient species don't use these criteria, so we excluded those. So only out of the 15 criteria, 8 were used in my research. And then for simplicity, we renamed the criteria A through H. And now for the results, I found that using the chi-square test of independence, you need to, we see that from the observed frequencies, so all the, for row A, for criteria A, which was morphology only, we see that out of a total of 1,349 species used morphology only, and 311 of those species were later determined as a synonyms. And I want to put your attention to F, G, and H, which include phylogenetic inference. And we see that, interestingly, they all, they don't lead to a synonym when combining phylogenetic inference. So then we took the expected frequencies from the observed frequency table and see that there was a violation for the chi-square test. So we couldn't test all of these because the chi-square assumes counts for greater than 5. So instead, we excluded those that were less than 5 and tested the chi-square test for just A, D, and E, and found that there was a significant statistically significance for A, D, and E. So what if we test those independently? So individually for criteria A, morphology only, we found that there was a statistic significance. And for criteria D, which was morphology with phylogenetic inference, we found significance. But for criteria E, which was morphology, micromorphology, and paleontology, we found that there was no statistic significance. So now that we've done them individually, what if we combine the original data, which had counts later than 5, and just combine those? So when combining those, there was still one cell counts less than 5, but the chi-square test that if the cells have 80% or more than 5, we can still use the chi-square test. So the chi-square test was still used when combining the data, and found that for this table, when combining B, C, D, E, F, G, that there was a statistic significance. So we want to see which cells contributed to that significance. And in order to find that, we used standardized residuals. And if a cell had an absolute value of greater than 1.96, then that cell contributed to that significance, so that we see E, F, and G was greater than 1.96, so that cell contributed to that significance. And also using, we wanted to double check using the p-values and the criteria, morphology, micromorphology, paleontology, morphology, micromorphology, phylogenetic inference, and morphology, paleontology, and phylogenetic inference was the criteria used that led to the statistic significance. So then another test to use is the Fisher's exact test. Since we had counts less than 5, we couldn't use the chi-square test for all of these criteria. So the Fisher's exact test was used. And this also looks at the relationship between two categorical variables, and the SPS software was used to use Fisher's exact test. And from SPSS, we found that morphology and morphology and phylogenetic was statistically significant, and this coincides with the chi-square test value shown here in yellow. And we also wanted to find out this effect size, the strength or the magnitude of the relationship with the criteria and species stabilities. So we used something called risk ratio and found out for morphology, the risk ratio was 2.7, so close to 3. And for morphology and phylogenetic inference, it was about 0.19, so close to 0.2. And if it was greater if it was greater than 1, we see that for morphology, it's more likely to be synonymized when the criteria used versus other criteria. And for morphology and phylogenetic inference, since it was less than 1, it was less likely to be synonymized when the criteria was used versus other criteria. So my results show that it showed that out of the 2 significant criteria, morphology was the only criteria that increased the risk of a species later being synonymized. So it was about 3 times more. And morphology with phylogenetic inference, we see that it had a protective effect, that it decreased the risk of being synonymized by almost 5 times. And when we see that, again, chi-square test, Fisher's test both conclude that morphology and morphology with phylogenetic inference were the only significant criteria that had a relationship. And then interestingly, again, I wanted to point out again that when phylogenetic inference were used, none of them or very few, such as criteria D, led to species being synonymized. But only, this was a limitation, only 23 species were used, only 6 species, 3 species, and 11 species were used. And this brings me to the limitations where there was a large discrepancy between the samples in each criteria. So that was one of the big limitations. And a future direction of the study is that, well, I want to conclude that adding phylogenetic inference with the other criterion seems to be the most promising. So we want to recommend using phylogenetic inference when discovering new species so that they won't be synonymized later. And a future study could determine which subtraits, such as within gross morphology, was it the flower, the leaf, was it better? Or for phylogenetic inference, which markers were better, excuse me, such as chloroplast, mitochondria, or nuclear DNA. And my acknowledgement slide, I want to show, and thank you.
Thank you. Lucky we shifted you the end there because you went 3 minutes over.
I'm sorry.
I had one question. I have no botanical background whatsoever, but is there a circularity in that my guess would have been that when a species is synonymized, that's done based on phylogenetic data, that what was called separate species is overruled and they're synonymized because phylogenetic data indicates that the original distinction was incorrect. So if that's the case, then it would have to be the case that any original designation using phylogenetic information is far less likely to be later over synonymized because that's the criteria used to synonymize or not. I don't know if I'm explaining that well, but
yes. Well, sorry. If I think for using phylogenetic inference, I mentioned that it is relatively the most objectives when using nuclear DNA, because you can construct a phylogenetic tree using morphology traits.
Sure, okay.
Yes, but it will be different because the traits using the tree with morphology is just looking at traits phenotypically, just looking at them, and just by looking at them morphologically, you can't determine if these are if they're like closely related or not. So instead we want to
convergent evolution or parallel evolution.
Yes, if they evolved closely related or if they were furtherly evolved.
Any other questions from the audience? We've got time because there is no, it's an empty slot, the last one in this session. If not, maybe join me in thanking all the speakers from this session. It's a very interesting and diverse set of talks. Thank you very much.
Thank you.
And I